Welcome to the deep dive. Today we're embarking on a journey into a topic that, well, it might seem purely academic at first glance, but its implications are just so deeply woven into global health, history, even our daily lives. Our mission for you, our dedicated listener, is to unlock the intricate world of cholera and other vibriosis. We're not just aiming for a surface level understanding here, no. Instead, we're diving profoundly into chapter 168 of our source material. We're going to dissect every detail, every concept, so you can achieve a true comprehensive mastery of this vital information. Think of it as gaining the kind of insight that really sets elite professions apart.
And that's precisely our objective, because to truly master this field, whether you're in public health, infectious disease research, or maybe you're just an intensely curious mind,
Yeah.
a deep understanding of these pathogens isn't just important, it's absolutely paramount. We'll trace their global impact, which has, literally shaped human civilization and continues to challenge healthcare systems worldwide. But we'll also drill down to the minute the molecular mechanisms by which they, well, wreak havoc in the body. This isn't just about piling up facts, it's about connecting the dots, recognizing the patterns, and appreciating the sophisticated interplay between these bacteria, our environment, and human physiology.
I'm really looking forward to that because our source material, chapter 168, it is a comprehensive, expert-level text. It's dense. It's packed with incredibly precise information. But that's exactly where the deep dive comes in. Our session today will distill that complexity, make it accessible, genuinely engaging, and I promise you, truly fascinating. We're going to pull out those surprising nuggets of knowledge, those aha moments that transform rote memorization into profound understanding.
Indeed. And it's crucial to highlight the broad scope we'll cover. While cholera, caused by Vibrio cholerae, is undoubtedly the classic and arguably most devastating disease within this chapter's focus, our source also meticulously delves into other significant bacterial infections. We'll begin with a brief but important look at Campylobacter, and then dedicate substantial time to a variety of other Vibrio species. Each of these has its own unique characteristics and distinct clinical presentations, so making that differentiation is a key part of our mastery today.
Okay, so before we plunge headfirst into the Vibrio family, our source material briefly touches on Campylobacter. It's distinct, yes, but it offers some crucial insights into this broader world of diarrheal diseases. Let's start there.
Okay, so when we examine Campylobacter enteritis, the general prognosis, thankfully, is quite favorable. The vast majority of patients, nearly all in fact, experience a full recovery. This can often happen spontaneously, without specific medical intervention, or be significantly aided by targeted antimicrobial therapy.
Right.
However, here's a critical insight. The few reported deaths from Campylobacter enteritis are almost exclusively attributable to profound volume depletion.
The dehydration.
Exactly. This underscores a fundamental, sometimes overlooked principle in managing severe diarrheal diseases. It's not typically the infection itself that's lethal, but the rapid, uncontrolled physiological consequences of massive fluid and electrolyte loss. The immediate priority must always be aggressive and timely hydration.
That distinction is incredibly important. So, uncontrolled dehydration is the primary killer, essentially. But are there any other complications, perhaps less obvious ones, that can arise from a Campylobacter infection? Our source mentions a couple that sound pretty serious, even if they're rare.
You're right to ask about those less common but significant sequelae. While general recovery is robust, a small percentage of patients can develop reactive arthritis. This is an intriguing autoimmune condition where the body's immune system, having fought off the Campylobacter infection, then mistakenly attacks its own joint.
Oh, wow. Leading to painful inflammation.
Yes, exactly. And even more rarely, but very seriously, Campylobacter infections are a known preceding event for developing Guillain-Barré syndrome or its variants.
Right, that's a neurological one.
Mm-hmm. A severe neurological disorder where the body's immune system attacks its own peripheral nerves. It leads to progressive muscle weakness, tingling, sometimes even temporary paralysis. These rare post-infectious complications really highlight how seemingly localized gut infections can trigger broader systemic immune responses with profound consequences.
That's fascinating and, yeah, sobering. So, does the severity of infection hold true across all Campylobacter species? Or are some more dangerous than others? Is it a uniform threat?
That's an excellent and crucial question, and it's where the nuances of the microbiology truly matter. Our chapter specifically highlights Campylobacter fetus as a significant outlier. Infections with C. fetus are associated with a much higher fatality rate compared to other Campylobacter species.
Much higher. Why is that?
Well, it isn't just about the organism itself being inherently more virulent in all cases. It partly reflects the population it typically affects. C. fetus frequently targets compromised hosts.
Ah, okay. People whose immune systems are already weakened.
Exactly. Individuals whose immune systems are compromised due to existing health conditions, making them profoundly vulnerable. For these patients, the prognosis is heavily dependent on the swift initiation of appropriate antimicrobial therapy. Delays in treatment can, unfortunately, lead to far more severe and often fatal outcomes in this specific context.
Can you elaborate on who these compromised hosts typically are?
Yeah.
And how does their pre-existing health status fundamentally alter the course of a Campylobacter infection, especially with C. fetus?
Certainly. An otherwise healthy host, someone without significant underlying medical conditions or an impaired immune system, will typically survive C. fetus infections without any long-term issues or sequelae. Their robust immune response, their physiological reserves, allow them to effectively combat the infection and recover fully.
But for compromised hosts,
The picture changes dramatically. This category includes individuals with a range of conditions: HIV/AIDS, cancer, especially those undergoing chemotherapy, the organ transplant recipients on immunosuppressive meds, severe malnutrition, or even advanced liver disease. Their diminished immune capacity means they struggle to clear the infection effectively.
right?
This puts them at a much greater risk of developing recurrent and/or life-threatening infections from various Campylobacter species. Their bodies are simply less equipped to withstand the direct assault of the bacteria and the physiological stress of a severe illness. This makes rapid and precise intervention absolutely critical for their survival.
That distinction is vital for understanding patient outcomes. Okay, now, let's transition to the true core of our deep dive today, the Vibrio genus. This is the primary focus of Chapter 168, and it's home to some truly globally impactful pathogens.
Indeed. The Vibrio species are responsible for a number of highly significant infectious syndromes. Among them, the one that stands out as the classic, the most devastating diarrheal disease, and the one that has truly shaped human history, is cholera, caused by Vibrio cholerae. Its name alone evokes a profound sense of historical suffering and ongoing public health challenges.
And when you reflect on its historical impact, it's truly staggering. We're talking about a disease responsible for no less than 7 global pandemics over the past 2 centuries. That's a history marked by immense suffering, countless deaths, and a pervasive fear that spread across continents. It's a stark reminder of how powerfully a single microorganism can shape not just public health, but also influence trade routes, migration, even political landscapes.
Absolutely, and despite that long and tragic history, the current relevance of epidemic cholera remains undeniable. It's far from a relic of the past. It continues to be a significant public health concern, particularly concentrated in the developing world today. Outbreaks still occur with alarming regularity, especially in regions grappling with poverty, poor sanitation, and severely limited access to safe drinking water. It just demonstrates that this ancient disease is, unfortunately, far from conquered.
Beyond cholera itself, our chapter describes a whole host of other vibrioses. What kinds of syndromes do these other Vibrio species cause? Is it always about diarrhea?
Not at all. While diarrhea is a common manifestation, other Vibrio species cause a remarkably diverse range of syndromes. These include various forms of gastroenteritis, yes, but also distinct soft tissue infections, which often arise from wound contamination in aquatic environments.
Oh, like from swimming with a cut.
Exactly.
Yeah.
And critically, some can cause primary sepsis, a severe life-threatening infection of the bloodstream. So, while V. cholerae is notorious for its diarrheal devastation, the Vibrio genus as a whole exhibits impressive versatility in its pathogenic potential. It attacks the body in multiple ways. Microbiologically, all Vibrio species share some key identifying characteristics. They are highly motile,
Meaning they move around actively.
yes, very actively, which is facilitated by one or more flagella, these whip-like appendages that propel the bacteria through liquid. They are also facultatively anaerobic, meaning they can survive and grow both with and without oxygen. Very adaptable.
Okay.
And importantly, they are characterized as curved gram-negative rods. That specific shape and staining characteristic helps in their lab identification.
So they're not just tiny aquatic acrobats, they're incredibly adaptable ones too, and their natural habitat, the environmental factors. That's absolutely crucial to understanding their spread, isn't it? It's not just about human transmission.
You've hit on a core ecological principle here. In their natural state, Vibrios are most commonly found residing in tidal rivers and bays. They thrive under conditions of moderate salinity. It's their perfect ecological niche. What's particularly critical is that they proliferate or multiply rapidly during the warmer summer months when water temperatures consistently exceed 20 degrees C.
20 degrees Celsius. That's a key threshold.
It's a very significant determinant of their growth. And as you might expect, there's a direct and profound correlation between these specific environmental conditions. Warmer water, increased salinity in certain aquatic environments, and the actual occurrence of human illness. When these conditions are met, the bacterial load in the environment significantly increases, directly raising the risk of human exposure and subsequent infection. This strong ecological link is fundamental to comprehending their epidemiology and, predicting outbreaks.
Let's get really precise now and define cholera. What exactly are we talking about when we use that term, especially in a clinical or public health context?
At its most precise, cholera is defined as an acute diarrheal disease. It has a terrifying, almost unparalleled capacity to cause profound and rapidly progressive dehydration. If left untreated, this can lead to death within a matter of hours. The sheer speed and volume of fluid loss are its defining and most feared clinical features.
And the chapter uses a very specific term for the most severe form, cholera gravis. That sounds as ominous as it is.
It is indeed. Cholera gravis represents the severe, life-threatening form of the disease. It has historically been, and tragically still is in many places, a much feared diagnosis, especially when it appears in its epidemic presentation. Imagine a patient losing multiple liters of fluid, up to 10 to 20 liters in a day, in just a few hours. It's a truly devastating condition that rapidly overwhelms the body's compensatory mechanisms, pushing it towards shock.
But fortunately, there's a critical silver lining here, isn't there? Despite its historical terror, there's a remarkably effective way to combat that historically high mortality. This is truly one of the most powerful insights.
Absolutely. And this is where the profound power of simple yet aggressive medical intervention comes into play. The good news is that prompt and aggressive fluid repletion, combined with essential supportive care, can dramatically prevent the historically high mortality associated with cholera. This is one of the most powerful and successful examples in medicine of how a relatively simple, accessible intervention replacing lost fluids can have an absolute profound impact on patient survival.
It's amazing. What kind of difference does it make in fatality rates?
Without this intervention, the fatality rate for cholera gravis can soar to over 50%. With it, when initiated promptly and correctly, it drops dramatically to less than 1%.
Less than 1%.
It's a triumph of basic physiology over a formidable pathogen.
So just to be crystal clear, the term cholera itself has a very specific modern meaning in medical and public health circles. It's not just any severe diarrhea, right?
That's a vital distinction. Historically, yes, the term cholera was sometimes applied quite broadly to any severely dehydrating secretory diarrheal illness, regardless of its specific cause. Could be infectious, even non-infectious sometimes. However, in contemporary medical and public health contexts, especially in our source material, cholera now specifically refers to disease caused by Vibrio cholerae serogroup O1 or serogroup O139.
O1 or O139. Why those two specifically?
Because this distinction is crucial. These are the only two serogroups currently known to possess true epidemic potential, meaning they are capable of causing the large-scale outbreaks and devastating pandemics we've witnessed throughout history. Any other Vibrio causing diarrhea is generally referred to as non-cholera vibriosis.
Now let's really dive deep into the classification and global journey of V. cholerae. How is this organism categorized, and what does that tell us about its potential to cause widespread disease?
Okay. V. cholerae is classified into over 200 serogroups. This distinction is based on the specific carbohydrate structures of their lipopolysaccharide, or LPS, O antigens. Think of these O antigens as unique molecular tags on the bacterial surface. What's absolutely crucial for our understanding is that while some non-O1 V. cholerae serogroups, meaning they don't clump together and antisert to the O1 group antigen, have occasionally caused sporadic, localized outbreaks of diarrhea. Serogroups O1 and O139 0139 are the absolute key players in large-scale disease.
01 and 0139 again.
Right. Until the emergence of 0139 in 1992, 01 was the exclusive cause of epidemic and pandemic cholera. So when we talk about the history of devastating widespread cholera, we're almost always talking about 01 or, more recently, 0139.
And even within that 01 serogroup, there are further distinctions, aren't there? Serotypes and biotypes. It's like a family tree of pathogens.
It is indeed, and these distinctions are significant for understanding its epidemiology. The O1 serogroup is further subdivided into two serotypes, known as Inaba and Ogawa, differentiated by subtle variations in their O antigen. And historically, there were two primary biotypes of V. cholerae O1, the classical biotype and the L-Tor biotype.
Classical and L-Tor.
Right. The classical biotype is now largely considered extinct, having been effectively displaced from its global dominance. The L-Tor biotype is the one currently responsible for the ongoing 7th global pandemic. It's a testament to bacterial evolution and competition.
That's truly fascinating, this idea of one biotype displacing another. So where does V. cholerae live naturally, and how does it make that leap to infecting humans? We know it's waterborne, but let's get into the nitty-gritty.
Its natural ecological niche is coastal saltwater and brackish estuaries. In these environments, it exists in a close, often symbiotic relationship with plankton, those microscopic organisms at the base of the marine food web. It can also survive, though less robustly, in freshwater environments, but only if there are adequate nutrients and sufficient warmth. Humans become infected, incidentally, typically by ingesting water or food that has become contaminated with bacteria. However, here's the critical point for its spread. Once infected, humans become essential vehicles for its dissemination.
They shed the bacteria.
Enormous numbers of bacteria in their feces, which then recontaminate water sources. The most common route of acquisition for humans is through the ingestion of water contaminated by human feces. Contaminated food, especially seafood or raw produce washed in contaminated water, also plays a very significant role in its spread. And importantly, for V. cholerae O1 or O139, there is no known animal reservoir.
That's crucial. No animal reservoir.
It's a crucial piece of information because it means that controlling human transmission and human-to-human environmental contamination is the primary, most effective target for preventing its spread.
That's an important insight. Now, what about the infectious dose? Is it easy to get cholera, or does it take a lot of bacteria? And what factors influence that dose? I hear it's not a simple number.
You're right, it's not a simple threshold. The infectious dose for V. cholerae, that is, the number of bacteria you need to ingest to get sick, is generally considered relatively high.
Relatively high. Meaning
a significant number of viable bacteria must survive the journey through your stomach and reach the small intestine to establish infection. However, this infectious dose is markedly reduced in specific circumstances, which helps explain why some people get sick more easily than others. For example, in hypochlorhydric persons, individuals with unusually low stomach acid, either naturally or due to medical conditions, the bacteria face less of an acidic barrier.
so stomach acid is protective.
Very protective. The same reduction in dose occurs in those who are using antacids, which intentionally neutralize stomach acid. And very importantly, the infectious dose is also reduced when gastric acidity is buffered by a meal.
Eating makes you more vulnerable.
Well, when you eat, the food helps to temporarily raise the pH of your stomach, allowing more bacteria to survive passage into the intestine. These factors highlight why the acidity of your stomach is a powerful, frontline natural protective barrier against cholera.
So our stomach acid is our first line of defense, which is fascinating. Who is typically most affected by cholera, and do we see any consistent seasonal patterns in outbreaks?
In areas where cholera is endemic, meaning it's consistently present, it predominantly affects children. It tends to be a pediatric disease there because older individuals typically develop some level of natural immunity from repeated exposures.
Makes sense.
However, when the disease is newly introduced into a population that has no prior immunity, like we saw in Haiti, it affects adults and children equally because no one has protective antibodies. In many endemic regions, there are often distinct cholera seasons that are closely linked to environmental conditions. Periods of high temperatures, heavy rainfall, and particularly flooding.
Right, creating conditions for spread.
Exactly. These conditions facilitate bacterial proliferation in water sources and enhance their spread through contaminated water networks. Despite these seasonal peaks, it's important to note that sporadic cases of cholera can occur year-round.
And geographically, it has a native home, doesn't it? A place where it's thought to have originated.
Yes, the scientific consensus is that cholera is native to the Ganges Delta on the Indian subcontinent. This region is widely considered its historical origin point, from which it has repeatedly spread across the globe.
And from that native home, it launched 7 global pandemics. Can you walk us through those, particularly the current one, the 7th, and what made it so unique?
Absolutely. Since 1817, humanity has tragically witnessed 7 global pandemics of cholera, each leaving a devastating trail of suffering and death across continents. The current 7th pandemic is particularly noteworthy and distinct. It began in Indonesia in 1961 and marked a significant shift.
What was the shift?
It was the first pandemic primarily caused by the L-Tor biotype of V. cholerae. This new biotype had certain characteristics, like longer survival in the environment and a greater propensity for asymptomatic carriage, that allowed it to spread in serial waves throughout Asia. It effectively displaced and largely extinguished the classical biotype, which is believed to have been responsible for all 6 previous pandemics.
Wow, so El Tor took over.
Completely. Then, in the early 1970s, L-Tor cholera erupted in Africa, causing major epidemics there before becoming a persistent endemic problem across the entire continent. This shift to L-Tor fundamentally altered the global epidemiology of cholera.
It's clear that it's still very much with us. What's the true global burden of cholera today, and how much of it, if any, goes unreported, potentially hiding the real scope of the problem?
That's a critical and often frustrating point for public health. While over 95% of cholera cases reported annually to the World Health Organization currently originate from Africa and Asia, as vividly illustrated on world distribution maps, the true global burden and precise distribution are largely unknown.
Why is that? Lack of testing.
Partly. The diagnosis, particularly in resource-limited settings, is often syndromic, meaning it's based on clinical symptoms rather than confirmed lab tests. Furthermore, many countries with endemic cholera simply lack the robust surveillance systems or, frankly, the incentive to report their cases accurately to the WHO.
So the numbers we see are likely underestimates.
Massive underestimates. It's conservatively estimated that there are potentially 1 to 4 million cases of cholera occurring yearly worldwide, yet only about 200,000 of those are actually reported. Similarly, of an estimated 20,000 to 140,000 deaths annually due to cholera, fewer than 2,000 are reported to the WHO. This vast disparity highlights a profound challenge in accurately assessing and responding to the global impact. It's a hidden epidemic within the reported figures.
That level of underreporting is truly staggering. And we saw cholera make a dramatic and tragic return to Latin America after a long absence, didn't we? The Haiti outbreak in particular was a harrowing example.
Yes, after a full century without cholera, the current pandemic strain reached Central and South America in 1991. It caused an initial explosive spread, affecting millions across the region in what was a profound public health crisis. While the burden has thankfully decreased markedly since that initial wave, the reintroduction served as a stark, painful reminder of its persistent global threat.
And Haiti in 2010.
A particularly devastating and widely studied example. This was a country with no recorded history of cholera, making the outbreak even more shocking and indicative of external introduction. Several lines of compelling evidence strongly suggest that cholera was likely introduced into Haiti by United Nations security forces from Asia.
That's incredibly controversial and tragic.
It is. And this event raised a very important possibility for future epidemic control, that asymptomatic carriers of V. cholerae, individuals who are infected and shedding bacteria but show no symptoms, can play a significant, often underestimated role in transmitting cholera over vast distances.
Spreading it across continents without even knowing.
Exactly. Inadvertently sparking new epidemics. The Haitian outbreak was catastrophic, ultimately involving over 800,000 individuals and resulting in thousands of deaths.
That's an incredibly tragic and telling example of how seemingly distant actions can have devastating local consequences. And we've seen other severe outbreaks in recent years, often tied to very difficult circumstances, haven't we? It seems cholera preys on vulnerability.
Unfortunately, that's a precise observation. The recent history of cholera has indeed been punctuated by severe outbreaks, particularly among impoverished or displaced populations, where public health infrastructure is either overwhelmed, damaged, or simply non-existent. A prime and horrifying example is the 2016 Yemen outbreak, which unfolded amidst a devastating civil war, massive population displacement, and a complete breakdown of essential public health systems.
Just a perfect storm for cholera.
Exactly. This tragic confluence led to an unprecedented scale of disease, with over 1.2 million suspected cases and thousands of deaths. It illustrates the dire consequences when public health measures collapse entirely. Other harrowing examples include the outbreak in Rwandan refugee camps in Goma, Zaire, back in '94, outbreaks in Zimbabwe from 2008-2009, and recurrent crises in South Sudan and the DRC in 2015. These cases powerfully emphasize the direct and undeniable link between large-scale cholera outbreaks and circumstances like war, natural disasters, or other humanitarian crises that lead to the profound breakdown of essential public health measures.
Like clean water and sanitation.
Precisely, those fundamental necessities.
It paints a very clear picture of just how vulnerable populations can become. What about in the U.S.? Do we see cholera here? And if so, how does it typically manifest? Is it a major concern?
Well, it's not an epidemic threat in the same way, but sporadic endemic infections due to V. cholerae O1 strains, specifically those related to the 7th pandemic strain, have actually been recognized along the U.S. Gulf Coast.
The Gulf Coast, like Louisiana and Texas.
Exactly, Louisiana and Texas primarily. These infections are typically linked to the consumption of contaminated, locally harvested shellfish, especially oysters. Occasionally, cases appear in U.S. locations far removed from the Gulf Coast, and these have usually been traced back to shipped-in seafood originating from the Gulf.
Ah, a reminder of our interconnected food supply.
Indeed. So, while not a widespread epidemic concern, localized transmission associated with seafood does occur.
And we also had the emergence of a new serogroup, O139, in the 1990s, which initially caused quite a stir, even concern about an 8th pandemic. What was its fate, and what's the lasting lesson from its appearance?
That's a crucial and fascinating part of the V. cholerae story. In October 1992, a large-scale outbreak of clinical cholera caused by this new serogroup, O139, erupted in southeastern India. This organism was a novel entrant onto the global stage. It appeared to be a derivative of the L-Tor O1 biotype, but with a distinct lipopolysaccharide, or LPS, and notably an immunologically related O-antigen polysaccharide capsule. This capsular presence was a significant difference because O1 organisms are not encapsulated.
So it had a capsule, unlike O1.
Right. After its initial rapid spread across 11 Asian countries, causing widespread concern about the onset of an entirely new pandemic, V. cholerae O139 has, interestingly, been almost entirely replaced by O1 strains once again.
So it didn't stick around.
Not really, not on a large scale. The clinical manifestations of the disease caused by O139 are indistinguishable from those of O1 cholera, same devastating watery diarrhea. However, a crucial public health and vaccine development point is this. Immunity acquired from prior exposure or vaccination to O1 does not protect against O139.
Ah, so prior immunity didn't work.
Correct. Which means the emergence of such a new serogroup requires distinct surveillance and potentially different vaccine strategies. Its temporary rise and subsequent decline illustrate the dynamic, ever-evolving nature of these pathogens.
Okay, let's unpack this. We've talked about the organism, its history, its spread.
Yeah.
But how does V. cholerae actually cause such devastating illness? What's the master plan, the biological weapon, behind the profound dehydration?
What's truly fascinating here, and central to understanding cholera, is that at its core, it is fundamentally a toxin-mediated disease. The characteristic profuse watery diarrhea, which is the most devastating and feared symptom, is due almost entirely to the action of a single, incredibly potent protein, cholera toxin, or CT.
Cholera toxin, CT.
Right. The V. cholerae organism elaborates or produces this toxin within the small intestine once it has successfully colonized that environment. It's a textbook example of a pathogen acting through a molecular hijacker.
So it's all about this cholera toxin. Can you break down its structure and how it works at a molecular level? What are the moving parts of this biological weapon?
Certainly. Cholera toxin has a very specific and, well, elegant structure. It's composed of a single monomeric enzymatic A subunit, that's the active or key part, and a pentameric binding B subunit, which is the lock-picking or delivery vehicle part.
A key and a delivery vehicle.
Okay? Think of it like a biological key designed to open a specific cellular door. The B pentamer is responsible for precisely binding to a particular lipid molecule called GM1 ganglioside. GM1 is found abundantly on the surface of intestinal epithelial cells.
So the B subunit finds the right door handle.
Exactly. This binding is absolutely critical because it enables the efficient delivery of the A subunit, the enzymatic destructive component, to its target inside the cell, specifically within the cell's cytosol. Without the B subunit to bind to GM1, the A subunit simply cannot gain entry to cause its damage.
So the B subunit is the perfect delivery vehicle. What happens once the A subunit is inside the cell? That's where the real molecular cascade begins, turning the cell against itself, right?
Precisely. Once inside, the activated A subunit, specifically a part of it called A1, performs a highly specific enzymatic reaction. It irreversibly transfers a molecule called ADP ribose from nicotinamide adenine dinucleotide to its specific target protein.
Its target protein being?
The GTP-binding regulatory component of adenylate cyclase. This is a crucial enzyme in cell signaling. This process, called ADP ribosylation, has a profound and permanent effect. It relentlessly upregulates the activity of adenylate cyclase.
Upregulates it, meaning it turns it way, way up.
Way up, and keeps it stuck in the on position. The direct and devastating result of this unregulated upregulation is the massive intracellular accumulation of very high levels of cyclic adenosine monophosphate, or cyclic AMP. This molecule is a crucial second messenger in normal cell signaling, but its uncontrolled buildup acts like a signal gone haywire, triggering a cascade of disruptive events.
So extremely high levels of cyclic AMP inside the intestinal cells. What does this all mean for the cell's normal function, and how does that translate into the profuse watery diarrhea? This is the core aha moment for understanding the dehydration.
This is indeed the crucial link, and it's elegant in its destructive simplicity. High cyclic AMP has a dual devastating effect on intestinal epithelial cells. In the villus cells, which are primarily responsible for absorption of nutrients and water in the small intestine.
Absorption, taking things in.
Right. High cyclic AMP powerfully inhibits the absorptive sodium transport system. This means sodium, and consequently water, cannot be effectively taken out of the intestinal lumen and back into the body. Simultaneously, in the crypt cells, which are typically involved in secretion of fluids.
Secretion, putting things out.
Exactly. High cyclic AMP actively stimulates and activates the secretory chloride transport system. This causes a massive pumping of chloride ions into the intestinal lumen. These two events, inhibition of absorption and relentless activation of secretion, led directly to a massive accumulation of sodium chloride, essentially common salt, within the intestinal lumen.
And then the watery diarrhea connection becomes crystal clear.
Exactly. Because water always moves passively to maintain osmotic balance, it follows the rapidly accumulating sodium chloride into the intestinal lumen. This results in an enormous amount of isotonic fluid accumulating there. When the sheer volume of this fluid exceeds the capacity of the rest of the gut, particularly the large intestine, to reabsorb it, the result is the profuse, unstoppable watery diarrhea characteristic of cholera. It's a relentless outpouring of fluid, literally a biological flush that can rapidly drain the body.
And without intervention, that fluid loss has truly devastating consequences.
Indeed. Unless that wasted fluid and electrolytes are adequately and promptly replaced, the patient rapidly progresses to shock, specifically hypovolemic shock, due to profound dehydration. Furthermore, the massive loss of bicarbonate from the body's fluid results in severe metabolic acidosis, which further compromises vital physiological functions and can quickly become lethal. It's important to mention that while the perturbation of the adenylate cyclase pathway by CT is the primary mechanism, CT also has additional, more subtle mechanisms by which it enhances intestinal secretion, maybe through prostaglandins or neural histamine receptors. It demonstrates that even in what appears to be a straightforward toxin-mediated disease, there can be multiple layers of complexity.
Beyond the toxin, what other critical virulence factors does V. cholerae possess, and how does it coordinate their production? Our chapter mentions the toxar regulon and something called quorum sensing, which sounds like bacterial communication.
These are absolutely key to understanding the bacterium's remarkable success in survival. First, there's the toxin co-regulated pilus, or TCP.
TCP, what does that do?
Think of TCP as tiny, sticky grappling hooks or adhesive structures. They are absolutely essential for V. cholerae to successfully attach to and multiply within, that is, to colonize the small intestine. It's specifically named toxin co-regulated because its synthesis is regulated in parallel with that of cholera toxin itself.
Ah, so they're made together.
Exactly. And this brings us to the toxar regulon. The production of cholera toxin, TCP, and several other crucial virulence factors are all coordinately regulated by a master regulator protein called toxar. This protein acts like a central command center, sensing various environmental signals in the gut-like pH or temperature. And then, through a cascade of other regulatory proteins, it precisely modulates the expression of genes coding for these virulence factors. This sophisticated system allows the bacterium to precisely control when and how it expresses its pathogenic arsenal, ensuring maximum impact at the right time.
So it's like a finely tuned symphony of virulence factors responding to cues. And you mentioned quorum sensing. That really does sound like bacterial communication.
It is, in essence, a form of bacterial communication that allows them to act as a collective. Quorum sensing refers to additional regulatory processes where bacteria respond to the density of their own population.
How does that work?
They constantly release small signaling molecules into their environment. When these molecules reach a certain concentration, indicating that a sufficiently large bacterial population, a quorum, has been achieved, they trigger coordinated changes in gene expression across the entire bacterial community. In V. cholerae, quorum sensing modulates virulence, meaning the bacteria don't launch a full-scale pathogenic attack until their numbers are sufficient to overwhelm the host's defenses. It ensures their collective efforts are impactful and efficient. It's a brilliant, coordinated survival and attack strategy.
That's truly a brilliant survival strategy for the bacteria. Our chapter also delves into genomic insights, shedding light on how V. cholerae acquires and shares its virulence factors. What does the genome tell us about this evolutionary arms race?
The V. cholerae genome is quite interesting. It's composed of 2 circular chromosomes, which is somewhat unusual for bacteria, many of which have only one. But what's particularly fascinating here, and absolutely critical to its rapid evolution and spread, is the pervasive role of lateral gene transfer.
Lateral gene transfer, meaning genes moving sideways between bacteria.
Exactly. It's a process where genetic material is transferred between bacteria horizontally, from one bacterium to another, rather than vertically from parent to offspring. The genes encoding cholera toxin, CTXA-B, are a perfect example of this. They are actually part of the genome of a bacteriophage, known as CTX.
A bacteriophage, a virus that infects bacteria.
Correct. And here's the twist. The receptor for this specific phage on the V. cholerae surface happens to be the very intestinal colonization factor we just discussed, TCP.
The grappling hooks.
The grappling hooks. This directly links the toxin's acquisition to the bacterium's ability to colonize the gut. Because CTXA-B is part of this mobile genetic element, this bacteriophage, it can be horizontally transferred between different V. cholerae strains, literally allowing bacteria to swap blueprints for their most potent weapon. This mechanism largely accounts for the historical emergence of new toxigenic V. cholerae serogroups.
It's like the virus carrying the master blueprint for the weapon, infecting new bacteria and giving them a new superpower.
That's a great analogy.
So bacteria are essentially swapping superpowers, or at least superweapons. And the chapter mentions pathogenicity islands in this context too. What are those, and what's their significance?
Yes, pathogenicity islands are another key concept. Many other important genes for V. cholerae pathogenicity, including those for TCP biosynthesis, accessory colonization factors, virulence gene regulation, are not scattered randomly across the genome. Instead, they are clustered together in distinct regions called V. cholerae pathogenicity islands.
So virulence genes grouped together.
Exactly. This similar clustering is also found in many other bacterial pathogens, suggesting a common evolutionary strategy. It's widely believed that these pathogenicity islands, much like the cholera toxin genes carried by the phage, were also acquired by bacteria through horizontal gene transfer from other organisms. A prime example given is V. cholerae O139, which you asked about earlier. It probably derived from an L-Tor-O1 strain that acquired the specific genes for O139 O antigen synthesis via this very mechanism. This genomic plasticity explains how V. cholerae can so rapidly evolve, acquiring new virulence capabilities and adapting, enabling its remarkable and often terrifying epidemic spread.
Moving from the microscopic mechanisms to the observable symptoms, let's talk about the wide range of clinical presentations patients experience with cholera. It's not always just a catastrophic picture, is it? It's a spectrum.
You're absolutely right. It's a remarkably broad spectrum. Individuals infected with V. cholerae O1 or O139 exhibit a surprisingly wide range of clinical manifestations. On one end, you have individuals who can be completely asymptomatic. They show no symptoms at all, even while actively shedding the bacteria.
Wow, asymptomatic carriers.
Yes. Or they may experience only very mild, self-limiting diarrhea that might not even be recognized as cholera. On the other end of the spectrum, however, patients can present with the sudden onset of explosive, life-threatening diarrhea, which is what we formally call cholera gravis. So the illness can range from virtually imperceptible to immediately fatal, highlighting the challenge in both surveillance and clinical management.
What factors influence how severe the disease becomes in a given individual? Is it just the infectious dose, or are there other host factors at play?
Several factors contribute significantly. These include, importantly, the level of pre-existing immunity a person has, usually developed through prior exposure in endemic areas. Interestingly, blood type plays a fascinating and not fully understood role.
Blood type. How so?
Persons with type O blood are at the greatest risk of developing severe disease if infected, while those with type AB blood appear to be at the least risk.
That's wild. Type O is riskier.
It seems so. It's an intriguing genetic link, though the mechanism is complex. Additionally, a person's nutritional status can significantly influence outcomes, with malnourished individuals often experiencing more severe illness.
So if you're a non-immune individual, someone who's never been exposed before, what does that typical progression look like? How quickly does it unfold?
In a non-immune individual, after a relatively short incubation period, typically between 24 and 48 hours, cholera characteristically begins with the sudden onset of painless watery diarrhea. And this isn't just mild looseness. This diarrhea can very quickly become extremely voluminous, leading to rapid and alarming fluid loss.
Painless but voluminous.
Exactly. Patients often experience frequent vomiting as well, which further exacerbates dehydration and electrolyte imbalance. The speed of onset and the sheer volume of fluid loss are truly distinguishing features.
And that volume loss can be truly extreme, can't it? I recall the numbers in the source are quite startling.
They are indeed. In severe cases, the volume of fluid loss can exceed an astonishing 250 milliliters per kilogram of body weight within the first 24 hours.
250 milliliters per kilogram. That's
For an average adult, that could mean losing 15 to 20 liters of fluid in a single day.
20 liters.
Without immediate intervention, if these fluids and electrolytes are not adequately and promptly replaced, the patient will rapidly progress to hypovolemic shock, a state where the circulatory system fails due to insufficient blood volume. And ultimately, death may ensue within hours. It's a race against time.
That's a truly terrifying amount of fluid. What are some of the key symptom characteristics that distinguish cholera from other diarrheal diseases? Are there any specific hallmarks that clinicians look for?
One of the key distinguishing features is that fever is usually absent.
No fever, typically.
Typically not. Unlike bacterial dysentery or many viral gastroenteritis cases, a patient with cholera gravis usually won't have a high temperature. However, muscle cramps, which can be quite severe and agonizing, are very common. These cramps are a direct, painful consequence of the significant electrolyte disturbances, particularly the loss of potassium.
And the stool itself has a very specific, almost iconic appearance, which our chapter refers to as rice water stool. Can you describe that in detail?
Absolutely. The stool in severe cholera has a characteristic appearance that is highly indicative. Once seen, rarely forgotten. It's described as non-bilious, meaning no bile gray and slightly cloudy fluid. It typically contains flecks of mucus, but critically there is no blood or pus present. It has a somewhat fishy but generally inoffensive odor.
And why rice water?
The resemblance is striking. It looks exactly like the water in which rice has been washed, hence the evocative name. This appearance is a direct result of the rapid, profuse, essentially clean fluid outpouring from the small intestine without significant inflammation or damage that would lead to blood. It's pure secretion.
And as the patient loses more and more fluid, how do the clinical finds parallel that progressive volume contraction? Our chapter mentions a table for degrees of dehydration, table 168-1.
The clinical signs are a direct reflection of the body's diminishing fluid reserves.
Yeah.
At losses of less than 5% of body weight, the patient might just experience increased thirst, maybe some restlessness. As dehydration progresses to 5–10% loss, more concerning signs appear. Postural hypotension.
Drop in blood pressure when standing up.
Right. Generalized weakness, tachycardia, increased heart rate as the body tries to compensate, and decreased skin turgor. The skin, when pinched, takes longer to return to normal. Mouth gets dry, tongue dry, maybe no tears.
Beyond 10% loss.
That's when the signs become truly life-threatening. Oliguria or anuria, severely reduced or absent urine output, signaling kidney trouble. Pulses become weak, thready, even absent. Sunken eyes are classic, sunken fontanelles in infants. The skin might get severely wrinkled, described as washerwoman skin, especially on hands and feet. Mental state deteriorates rapidly. Somnolence, profound lethargy, eventually coma.
So the body is essentially shutting down system by system. What are the major complications, and crucially, can they be prevented if treatment is initiated promptly?
What's absolutely crucial to understand, a key takeaway, is that the complications of cholera derive almost exclusively from the profound effects of volume and electrolyte depletion. The primary and most serious complication is acute renal failure, specifically acute tubular necrosis, resulting directly from prolonged hypovolemic shock and inadequate blood flow to the kidneys.
Why?
But, and this is the hopeful part, if the patient is adequately and promptly treated with sufficient fluid and electrolyte replacement, These severe complications are entirely averted. The disease process itself, the shedding of bacteria, the toxins action, it's self-limited. It resolves on its own in a few days once fluid balance is restored and maintained.
Self-limiting. That's huge.
It underscores the immense power of timely and aggressive rehydration as the life-saving intervention.
And what would standard laboratory findings look like for a cholera patient? What clues would a clinician see in the blood work?
Lab data typically provides several key clues. You'd likely see an elevated hematocrit in non-anemic patients due to hemoconcentration, the blood getting thicker from fluid loss. There's often a mild neutrophilic leukocytosis, an increase in certain white blood cells. Blood urea nitrogen, BUN, and creatinine levels will be elevated, consistent with pre-renal azotemia, the kidneys being underperfused due to dehydration.
Okay.
Electrolytes like sodium, potassium chloride might appear normal initially in the serum, but a critical finding is a markedly reduced bicarbonate level, usually below 15 millimol.
Low bicarbonate means?
Severe metabolic acidosis. There will also typically be an elevated anion gap due to increases in serum lactate, protein, and phosphate. Correspondingly, the arterial pH is usually low, often around 7.2 or even lower, confirming the acidosis. These labs paint a clear picture of the physiological crisis.
Now let's turn to how this critical disease is identified, especially given its rapid progression. How is cholera diagnosed in the field or in a lab?
Clinical suspicion for cholera should be very high under specific circumstances. It should be suspected immediately when a patient, especially someone 5 years or older, develops acute, profuse, watery diarrhea in an area known to have ongoing cholera transmission.
Okay, known cholera area is a big flag.
Yes. The other critical scenario is if a patient develops severe dehydration or, tragically, dies from acute watery diarrhea, even if they are in an area where cholera is not generally known to be present. These are the red flags that demand immediate diagnostic protocols and public health response.
So clinical suspicion is the starting point, especially in an outbreak setting. How do you confirm the diagnosis definitively in the lab?
The definitive confirmatory diagnosis relies on the direct identification of V. cholerae in the patient's stool. It's crucial to emphasize that the organism must be specifically sought by the laboratory.
Meaning you have to ask for it.
Yes. Routine stool cultures, if not specifically instructed to look for Vibrio, might not use the appropriate selective media to isolate it. Communicating the clinical suspicion to the lab is key.
What are the best methods for detecting it in the lab? From quick field tests to more sophisticated culture methods.
For rapid initial detection, particularly in endemic or outbreak areas with sufficient experience, V. cholerae can actually be detected directly by dark field microscopy on a fresh stool wet mount. This lets you see the bacteria's characteristic darting motility.
Right, motility.
Furthermore, if you suspect O1, its specific serotype can be discerned by an immobilization test using O1 antiserum. The bacteria stop moving in the presence of the antibody. For definitive culture, selective media are paramount, like teroxalate tellurite gelatin, TTG agar, or thiosulfate citrate bile salt sucrose, TCBS agar.
TCBS, I've heard of that one.
Right. These inhibit many other common gut bacteria, letting Vibrio grow preferentially. If there's a delay in processing the sample, specialized transport media like Carrie Blair or alkaline peptone water enrichment medium should be used to keep the organism viable.
And what about biochemical confirmation? Is that always necessary, especially in resource-limited settings?
In endemic areas where cholera is common, there's actually little need for extensive biochemical confirmation. The clinical picture, rapid tests, or simple culture on selective media is often sufficient for immediate treatment and public health action. However, in places where V. cholerae is uncommon, these biochemical tests become more worthwhile for definitive confirmation. Standard biochemical testing used for Enterobacteriaceae usually works. A key identifying trait for all Vibrios is that they are oxidase positive.
Oxidase positive. Got it. And are there any modern rapid diagnostic tools for field use that have made a difference?
Yes. Thankfully, significant advancements have been made. Point-of-care antigen detection cholera dipstick assays are now commercially available.
Dipsticks, like a pregnancy test.
Similar concept, yeah. These are invaluable tools for use directly in the field, especially in remote areas or during humanitarian crises where labs are lacking or overwhelmed. They allow for quick, on-the-spot presumptive diagnoses to guide immediate public health responses and initiate treatment without waiting for lab results. That can be life or death in cholera.
Now, we've talked about the profound and rapid progression of cholera, the terrifying speed of dehydration. So let's address the single most crucial intervention for this disease. It's treatment. This is where lives are saved.
Absolutely. What's absolutely paramount here, the immediate priority, the absolute first step in treating cholera is fluid resuscitation. This is the bedrock of care. Death from cholera, as we've sensibly discussed, is primarily due to hypovolemic shock from massive fluid loss. Therefore, treatment first and foremost requires aggressive, often massive and rapid, fluid resuscitation and ongoing fluid management. Without this critical intervention, all other treatments are largely futile.
And treatment is guided by the patient's level of dehydration, as outlined in those tables in our source. What's the dual goal here for fluid management?
The primary goal is twofold and needs pursuing simultaneously. First, rapidly restore euvolemia, normal circulating blood volume, pulling the patient out of shock. Second, and equally important, continuously maintain adequate hydration by replacing the ongoing fluid losses from persistent diarrhea and vomiting. The volumes and routes are precisely tailored to the degree of dehydration, age, and weight, following established protocols.
The chapter really emphasizes the remarkable power of oral rehydration solution, or ORS. How does ORS work so effectively, even when cholera toxin is actively wreaking havoc in the gut? It seems counterintuitive.
This is truly a marvel of modern medicine and a testament to understanding basic physiology. What's remarkable about ORS is that it takes advantage of a specific physiological mechanism, the hexosne plus co-transport mechanism.
Glucose-sodium co-transport.
Mainly, yes. This system remains remarkably intact and functional even when cholera toxin is active. Here's how it works. The glucose in the ORS solution is actively transported across the gut mucosa, and critically, sodium ions, Na+, are co-transported along with it, like a piggyback ride. As sodium moves into the body, chloride ions, Cl, and most importantly, water molecules passively follow, drawn by the osmotic gradient. So, even though the toxin is causing massive secretion, this co-transport system allows for active absorption of vital electrolytes and, consequently, water from the gut back into the bloodstream.
It's like a physiological loophole.
Exactly. A bypass that allows rehydration despite the toxin's effects, literally saving lives by reestablishing fluid balance.
That's a truly ingenious mechanism, and it's incredible how such a simple solution can have such a profound impact. Our source even gives a recipe for homemade ORS, as well as mentioning the WHO's recommendations.
Yes, ORS can be conveniently made by adding safe water to prepackaged sachets of salts and sugar. But where those aren't available, a simple, life-saving homemade alternative can be prepared. It involves combining about half a teaspoon, roughly 3.5 grams of table salt, with either 50 grams of pre-cooked rice cereal or 6 level teaspoons of table sugar.
6 teaspoons of sugar or the rice cereal.
Right. All meticulously mixed into 1 liter of safe drinking water. This simple formulation can be a lifesaver. It's also crucial to encourage potassium intake from bananas or green coconut water, maybe especially if it's not in the ORS, as lots of potassium is lost.
Good tip.
The WHO now widely recommends low osmolarity ORS for dehydrating diarrhea of any cause. It's shown to be superior, particularly in cholera, due to better absorption. And if available, rice-based ORS is considered superior to standard ORS in cholera treatment, offering even better clinical outcomes.
So ORS is the front line, and it can even be administered via a nasogastric tube. But when is intravenous fluid replacement absolutely essential? And what's the best choice of fluid in those critical scenarios?
While ORS is truly a miracle, for individuals with severe dehydration, optimal management absolutely includes rapid IV fluid and electrolytes. This is especially true because profound metabolic acidosis, with pH often below 7.2, is common and dangerous in this group.
So which IV fluid?
Ringer's lactate is considered the best choice among commercially available solutions. It's preferred because it contains lactate, which the liver metabolizes to bicarbonate, directly helping to correct the severe acidosis.
And if Ringer's isn't available?
Normal saline is an acceptable alternative, but it must be used with additional potassium supplements, preferably given orally, as saline doesn't have enough potassium to replace the losses.
And how quickly do you need to administer these IV fluids? Is it a slow drip, or is speed of the essence? And for how long might it be needed?
Speed and volume are absolutely critical. The total fluid deficit in severely dehydrated patients, often more than 10% of body weight, can and should be safely replaced within the first 3 to 4 hours.
3 to 4 hours for the whole deficit.
Yes. And for patients in shock, half of that total volume can often be given within the very first hour. This rapid initial bolus is vital. During rapid rehydration, transient muscle cramps and even tetany are common due to electrolyte shifts, but they typically resolve. Once the patient is awake, can drink ORS, and has a decent pulse, you can transition to oral therapy to maintain hydration. However, patients with continued large volume diarrhea may need prolonged 5E treatment to match ongoing losses. Severe hypokalemia, low potassium can develop and needs potassium replacement, IV or oral. In settings with inadequate staffing for close monitoring, the oral route for rehydration and potassium is often considered safer than IV due to lower risk of fluid overload or imbalances.
So fluids are the priority, the absolute game changer. Beyond that, what role do antibiotics play in cholera treatment? Are they absolutely necessary for a cure, or do they serve a different purpose?
It's important to clarify a common misconception. Antibiotics are not necessary for curing cholera. The primary life-saving intervention is, unequivocally, fluid replacement. However, antibiotics do play a very valuable adjunctive role. If the organism is susceptible, antibiotic use significantly diminishes both the duration and the total volume of fluid loss. The patient recovers faster, loses less fluid. Furthermore, antibiotics hasten the clearance of the organism from the stool, reducing the period of infectivity and helping curb further transmission.
Okay, so they shorten illness and reduce spread.
Exactly. Therefore, adjunctive antibiotics should be administered to patients with moderate or severe dehydration due to cholera. They improve outcomes and aid public health control.
So they're not curative on their own, but they clearly make a big difference. What are the specific antibiotic choices, and what are the crucial considerations regarding antibiotic resistance, which seems to be a growing problem?
For choices, macrolides like erythromycin are common. The adult dose is 250 milligrams orally 4 times a day for 3 days. Children get 12.5 kilograms per dose, same schedule. Azithromycin is another excellent macrolide, often preferred for convenience. A single 1 gram dose for adults or a single 20 kilograms for children.
Single dose is much easier.
Definitely. Tetracyclines were historically first line, but unfortunately, widespread resistance is now reported in many endemic areas. If local susceptibility is confirmed, tetracycline itself can be used, or doxycycline as a convenient single-dose option.
But only if it's known to be susceptible locally.
Crucially. Similarly, increasing resistance to fluoroquinolones like ciprofloxacin is being reported. So again, local susceptibility patterns are key. If susceptible, ciprofloxacin can be used. It's absolutely crucial for clinicians to know the local resistance patterns of V. cholerae to guide appropriate and effective antibiotic choice. An ineffective antibiotic wastes time and resources.
And are there any other adjunctive therapies that have proven beneficial, particularly for young children with cholera?
Yes, absolutely. And this is a simple yet very impactful intervention: oral administration of supplemental zinc. Numerous studies show it's associated with decreased volume and severity of diarrhea in young children, including those with cholera.
Zinc helps with the diarrhea.
It does. It's a very important low-cost intervention that improves recovery. For children under 6 months, the recommendation is 10 milligram oral zinc daily for 10 days. For 6 months to under 60 months, it's 20 milligram daily for 10 days. Integrating zinc significantly aids recovery and reduces the illness burden on the child.
Beyond immediate treatment, the long-term battle against cholera really focuses on prevention, doesn't it?
Yeah.
What are the core public health measures that can significantly reduce its incidence on a population level?
Precisely. Prevention is the ultimate goal, and it rests on fundamental public health pillars. The core measures are ensuring provision of safe, clean water for drinking and sanitation, establishing adequate facilities for sanitary disposal of human feces.
Breaking that fecal-oral cycle.
Exactly. Improving nutrition across the population, as healthier individuals are less susceptible, and careful attention to food preparation and storage practices within households. When these measures are adequately implemented and sustained, they can profoundly reduce the incidence of cholera by disrupting its primary transmission routes.
And it's not just about local measures, but also preventing international spread. The Haiti outbreak was a stark and tragic reminder of just how easily this pathogen can cross borders.
It was indeed a painful lesson. Rigorous precautions should be taken to prevent international spread via infected and potentially asymptomatic persons traveling from endemic to non-endemic regions. The Haiti outbreak, likely introduced by UN forces from Asia into a previously cholera-free country, is a powerful illustration of how global travel can inadvertently facilitate long-distance transmission.
Sparking new epidemics in vulnerable populations.
Precisely. This makes international cooperation, surveillance, and rapid response vital.
For decades, there's been a lot of effort put into developing cholera vaccines. How have those developed over time, and what types are available now for use?
Much scientific and public health effort over decades has indeed been devoted to developing effective cholera vaccines, with a particular focus on oral vaccine strains. The aim is to elicit strong mucosal immune responses directly in the gut, where the infection takes hold.
Makes sense to target the gut.
Right. To maximize these responses, two primary types of oral cholera vaccine have been developed and are widely used: oral killed vaccines and oral live attenuated vaccines. Each has its own advantages.
And what are the specifics of these oral killed cholera vaccines? What are the key differences between them?
Currently, 3 oral killed cholera vaccines have been pre-qualified by the WHO, making them available internationally. One type is BIVWC, marketed as Shunchello, Uvicol, and Uvicol Plus. This contains both biotypes and serotypes of B. cholerae O1 and the O139 serogroup. Notably, it does not include supplemental cholera toxin B subunit.
Okay, BIVDOVC has O1 and O139, no extra B subunit.
Correct. The other major type is WCRBS, widely known as Dukoral. This vaccine contains both biotypes and serotypes of V. cholerae O1, but it is supplemented with 1 milligram of recombinant cholera toxin B subunit per dose.
Dukoral has O1 only, but adds the B subunit. Why add that?
The B subunit is purified and included to potentially enhance the immune response by mimicking the binding component of the natural toxin, hopefully inducing stronger antitoxin antibodies.
How are these killed vaccines administered? And how effective are they in real-world settings? Do they provide long-lasting protection?
They're typically given as a 2 or sometimes 3 dose regimen, usually separated by about 14 days for optimal immune development. They generally provide good protection, around 60% to 85% for the first few months.
And longer term, boosters needed.
For WCRBS, boosters are recommended after 2 years for those 6 and older, and after 6 months for kids 2 to 5 to maintain protection. For BIVWC, used more in endemic settings, recommendations are still evolving. But one study in Kolkata showed about 60% protection over 5 years, though lower, around 40%, for kids under 5. In outbreak situations, even a single dose of BIVWCS can provide some immediate protection, though maybe not in the youngest children.
Single dose helps in outbreaks. That's useful.
Yes. Models also predict significant herd immunity when vaccination coverage exceeds 50%. And crucially, these killed vaccines are safe in HIV-infected populations.
Beyond the killed vaccines, there are also oral live attenuated vaccines in development, which sound particularly promising, especially the idea of single-dose protection.
Yes, oral live attenuated vaccines for V. cholerae O1 are a significant and exciting area. These strains share a critical feature. They've been genetically engineered to lack the cholera toxin genes.
So they can't cause the severe disease?
Exactly. They can colonize the gut and elicit an immune response, but without the devastating toxin. One such vaccine, CBD103-HGR, known as Vaxchora, is FDA approved for travelers to cholera endemic regions.
How effective is Vaxchora?
Quite impressive. 90% efficacious against severe cholera 10 days post-vaccination in volunteer studies, and still 80% after 90 days. It's approved for ages 2 to 64. No booster recommendations currently, suggesting potentially long-lasting effect. A major advantage is the potential for robust protection after just a single oral dose. That's huge for mass vaccination campaigns during outbreaks.
Definitely simplifies logistics.
Immensely. Other candidate strains from L-Tor and O139I cholerae have also been tested, expanding the pipeline.
Are there other vaccine types being explored beyond these oral options, perhaps using different technologies?
Yes, the R&D pipeline continues to diversify. Conjugate cholera vaccines are under development, linking polysaccharides to carrier proteins for a stronger, longer-lasting response, especially in young children. Additionally, subunit cholera vaccines, focusing on specific antigenic components rather than the whole organism, are being explored.
How are all these prevention tools being integrated into global control strategies? It sounds like it's more than just vaccines.
You're absolutely right. It's a multi-pronged approach. WHO explicitly recommends incorporating cholera vaccination into comprehensive control strategies, alongside robust efforts for safe water and sanitation. An international stockpile of oral killed vaccine exists for rapid outbreak responses.
A global stockpile ready to go.
Yes. Furthermore, a major global strategy on cholera control was launched in 2017. It's a country-by-country approach. Ambitious aims: reduce cholera deaths by 90% and eliminate cholera in up to 20 countries by 2030.
Wow, 90% reduction and elimination in 20 countries by 2030.
It's ambitious. The integral components are advancing water, sanitation, and hygiene WWASH programs alongside strategic use of cholera vaccines. Between 2016 and 2020 alone, over 64 million doses were requested from the global vaccine stockpile, and over 33 million shipped, showing the commitment.
Now, let's broaden our scope beyond V. cholerae to other human pathogens within the diverse Vibrio genus. What are the general characteristics of these non-cholera Vibrios, and where do they typically reside?
What's truly fascinating here is how incredibly abundant these non-cholera Vibrios are in coastal waters throughout the world. They are a natural part of these marine ecosystems. Critically, they can reach very high concentrations in filter-feeding mollusks, oysters, clams, mussels.
Shellfish concentrate them.
Exactly. These act like living filters, concentrating the bacteria, making them a significant vector for human infection. As a result, human infection commonly follows ingestion of contaminated seawater, maybe while swimming, or more frequently from consuming raw or undercooked shellfish. The strong link between seafood, especially raw shellfish, and non-cholera vibriosis is a key point in our source.
How are these non-cholera Vibrios identified in a typical microbiology lab? Are they difficult to culture?
Fortunately, most non-cholera Vibrios can be readily cultured on standard lab media like blood agar or McConkey agar. These media contain enough salt for these halophilic species salt-loving.
Halophilic, okay.
In the lab, distinguishing between the various species is typically done using a battery of standard biochemical tests, assessing their unique metabolic characteristics.
And which are the most important species among these non-cholera Vibrios, the ones that pose the most significant threat?
Based on frequency and severity, the two most important species are Vibrio parahemolyticus and Vibrio vulnificus. These cause the most significant public health concern and the majority of severe non-cholera vibriosis cases.
What is their overall impact in the U.S.? Do we have a sense of the scale of illness they cause annually?
Yes, the impact is quite substantial, even if not pandemic scale. In the U.S. alone, vibriosis, the collective term, is estimated to cause around 80,000 illnesses and, tragically, about 100 deaths annually. This underscores their significant ongoing public health burden requiring vigilance.
So we're talking about two major types of syndromes caused by these non-cholera Vibrios. Can you break them down for us? Is it always a gut infection?
Generally, two major categories. First is gastrointestinal illness, primarily caused by V. parahemolyticus, but also non-O101 through V. cholerae, V. mimicus, V. fluvialis, V. holosae, and V. furnaceae. These typically manifest as diarrhea. The second major category is soft tissue infections, commonly caused by V. vulnificus, V. alginolyticus, and V. damseli.
Skin and muscle infections.
Usually, yes. Following exposure of a wound to contaminated water, it's also critical that V. vulnificus is somewhat unique. It can also cause primary sepsis, severe bloodstream infection, particularly in certain compromised individuals, even without a prior wound infection.
Let's focus first on the Vibrio species primarily causing gut issues. And the big one here, the leading cause of seafood-borne bacterial enteritis, is Vibrio parahemolyticus.
Parahemolyticus is indeed widespread in marine environments, and like many Vibrios, it is halophilic, thrives in salt. It holds the dubious distinction of being the leading seafood-borne bacterial cause of enteritis worldwide. Its historical significance was highlighted in Japan in 1953, implicated in 24% of enteritis cases in one study linked to eating raw seafood.
And what's its prevalence like in the U.S., and are we seeing an increase in reported cases?
In the U.S., common source outbreaks have consistently been linked to undercooked or improperly handled seafood or foods contaminated by seawater. What's particularly concerning is that since the mid-1990s, the incidence of V. parahemolyticus infections has increased significantly in several countries, including the U.S.
Incidence is increasing. Why?
Specific serotypes, namely O3K6, O4K68, and O1K untypable, which are genetically related, account in part for this observed increase, suggesting successful clonal expansion. This rising trend is clear in surveillance data. In 2014, it accounted for almost half of all Vibrio isolates reported in the U.S.
Half of all Vibrio isolates. Wow.
It highlights its growing importance as a foodborne pathogen.
How does V. parahemolyticus actually cause disease in the gut? What's its pathogenic mechanism, and are there particular virulence factors at play?
Its enteropathogenicity is strongly associated with its ability to cause hemolysis, red blood cell destruction via a protein called thermostable direct hemolysin, or VP-TDH. While the precise mechanisms of diarrhea aren't fully defined like CT, we know type 3 secretion systems T3SS1 and T3SS2 are crucial.
Those molecular injectors again.
Exactly. They directly inject toxic bacterial proteins, effectors, into host cells, disrupting processes. The activity of at least one T3SS is required for intestinal colonization and full virulence in animal models. So clinical suspicion for V. parahemolyticus should always be raised in any acute diarrhea linked to recent seafood consumption or sea exposure.
So seafood and sea exposure are the big clues. What are the typical clinical presentations of V. parahemolyticus infection? Does it also cause rice water stool?
Not typically the classic rice water stool. There are generally two distinct gastrointestinal forms. The more common one, nearly all North American cases, is watery diarrhea, abdominal cramps, nausea, vomiting. About 25% also have fever and chills.
Fever and chills can help differentiate it from cholera.
Yes. Incubation is 4 hours to 4 days. Symptoms last a median of 3 days. The less common, more severe presentation is dysentery, severe abdominal cramps, nausea, vomiting, and stools that are bloody or mucoid. Beyond the gut, rare cases of wound infection, otitis, ear infection, and very rarely sepsis.
How are these infections treated, and what's the prognosis for patients? Is it typically severe?
Most cases of V. parahemolyticus gastrointestinal illness are self-limited. They resolve on their own in a few days if fluid balance is maintained.
Fluids again.
Fluid replacement is paramount, always stressed. Antimicrobial agents may be beneficial in moderate or severe disease or in immunocompromised patients. Doxycycline, fluoroquinolones, macrolides, third-gen cephalosporins are options. Prognosis is generally excellent for healthy individuals. Deaths are extremely rare. Severe infections and fatalities are primarily linked to underlying diseases: diabetes, liver disease, iron overload states, immunosuppression, highlighting vulnerability.
Let's move on to non-010-139, non-cholera V. cholerae. These sound tricky to distinguish from the classic cholera strains. How do labs tell them apart, and what's their clinical significance?
They can indeed be tricky, a highly heterogeneous group. While they can't be distinguished from O1 or O139 by routine biochemical tests, a key difference is they do not agglutinate clump in O1 or O139 antiserum. That confirms they aren't the epidemic strains.
So the antibody test is key.
Right. Generally, non-O10139V cholerae strains do not produce cholera toxin and don't cause large diarrheal epidemics. However, they have caused well-studied foodborne gastroenteritis outbreaks and sporadic cases of otitis media, wound infection, and bacteremia. Like other Vibrios, they're widely distributed in marine environments. U.S. cases often linked to raw oysters or recent travel. The broad clinical spectrum is likely due to their heterogeneous virulence attributes. They vary in what makes them pathogenic.
What's their incidence like in the U.S., and what are the typical symptoms for gastroenteritis caused by these non-010-139 strains?
In the U.S., about half of all non-010-139V cholerae isolates come from stool samples, confirming their role in GI illness. Typical incubation for gastroenteritis is short, less than 2 days. Illness lasts about 2 to 7 days. Stools can vary. Copious watery, similar to cholera but less extreme or partly formed, less voluminous, sometimes bloody mucoid. Variability is key here.
So not always the classic cholera picture.
Right. Diarrhea can still be severe enough for dehydration if not managed. Many cases also include cramps, nausea, vomiting, fever.
And for treatment, is it similar to cholera with the fluids? What about antibiotics for these non-010-139 strains, especially for extraintestinal infections?
For seriously dehydrated patients, treatment is similar to cholera, emphasizing oral or IV fluids. However, for the gastroenteritis itself, the value of antibiotics is less clear, often not essential, as many cases are self-limiting. Where antibiotics become unequivocally important is for extraintestinal infections.
Wound infections, ear infections, bacteremia.
Exactly. These commonly follow occupational or recreational seawater exposure. About 70% are wound infections, 10% otitis media, 20% bacteremia, especially in liver disease patients. These extraintestinal infections should be treated with antibiotics due to potential severity. Specific guidance is limited due to heterogeneity, but most strains show in vitro sensitivity to tetracycline, ciprofloxacin, and third-gen cephalosporins, providing options.
Now, let's turn our attention to a truly concerning group: Vibrio species that cause infections primarily outside the gut, often with much more severe and life-threatening implications. The first and most concerning here is Vibrio vulnificus.
V. vulnificus infection is indeed rare, but it is unequivocally the most common cause of severe Vibrio infections in the United States. It carries a very high mortality rate demanding immediate attention. Like most Vibrios, it proliferates significantly in warm summer months and absolutely requires a saline environment, tied to coastal areas.
Where and when is it most common in the U.S.?
U.S. infections typically occur in coastal states between May and October, aligning with warmer water temps. Most commonly affect men over 40, often with underlying health conditions. The name vulnificus itself is Latin for wound maker, reflecting one key syndrome.
And our chapter clearly outlines 2 distinct syndromes for V. vulnificus. What are they, and what are their defining characteristics?
V. vulnificus is famously linked to two very distinct syndromes. First is primary sepsis, a severe, rapidly progressing bloodstream infection. Usually occurs in patients with underlying liver disease, especially cirrhosis, or conditions like hemochromatosis, iron overload enhances virulence
Iron overload helps the bacteria.
significantly. The second syndrome is primary wound infection. Generally affects people, sometimes even without underlying disease, caused by direct contact of a wound with seawater. Some authors also suggest it can cause gastroenteritis, though less common or severe than its other manifestations. The organism has potent virulence attributes: a capsule-resisting phagocytosis and serum killing, a cytolysine-damaging host cells. And critically, its virulence is considerably increased under iron overload conditions, explaining its propensity to infect hemochromatosis patients.
Let's delve into the primary sepsis in more detail. Who is most at risk, and what does that progression look like clinically? It sounds terrifyingly rapid.
Primary sepsis most often develops in patients with cirrhosis or hemochromatosis, but it can also affect those with hematopoietic disorders, chronic renal insufficiency, immunosuppressive meds, heavy alcohol use, rarely even those with no known disease. After a median incubation of just 16 hours, incredibly rapid, the patient develops malaise, chills, high fever, profound prostration.
16 hours, that's fast.
Frighteningly fast. About 1/3 develop hypotension, often upon admission, signaling impending shock. A hallmark are the distinctive cutaneous manifestations, developing in most cases within 36 hours, usually on extremities lower more than upper.
What do these skin lesions look like?
Characteristic progression starts with diffuse erythematous patches, then rapidly evolve to ecchymoses, bruises, vesicles, small blisters, then blee, large fluid-filled blisters, often hemorrhagic. Sepsis with these hemorrhagic bullous skin lesions strongly suggests V. vulnificus, especially with seafood coastal exposure. Necrosis and sloughing may occur. Aggressive.
And what about the lab findings, and critically, the prognosis for V. vulnificus sepsis? That mortality rate you mentioned earlier is incredibly high.
Labs often show a complex picture: leukopenia, low white cells, more often than leukocytosis, thrombocytopenia, low platelets, elevated fibrin split products indicating clotting issues. V. vulnificus can be readily cultured from blood or cutaneous lesions for diagnosis. However, the mortality rate approaches 50%. Most deaths from uncontrolled sepsis, multi-organ failure, shock.
50% mortality.
Prompt aggressive treatment is absolutely critical. Immediate empirical antibiotics upon suspicion, aggressive surgical debridement of necrotic tissue, comprehensive supportive care. In vitro, it's typically sensitive to tetracycline, fluoroquinolones, third-gen cephalosporins. Animal models and clinical experience often suggest a fluoroquinolone or R combination of tetracycline and a third-gen cephalosporin for septicemia. Urgency cannot be overstated.
And what about the V. vulnificus associated soft tissue infection, the wound maker aspect? How does that manifest, and what's the prognosis compared to sepsis?
The soft tissue infection typically complicates a fresh or old wound contacting seawater. The patient may or may not have underlying disease, though compromised hosts are higher risk. Short incubation, 4 hours to 4 days, mean 12 hours, onset. Rapid swelling, erythema, redness, often intense, disproportionate pain around the wound,
Intense pain is a key sign.
often. Followed by rapidly spreading cellulitis, sometimes with vesicular, bullous, or necrotic lesions, similar to sepsis, but initially localized. Metastatic events are uncommon, unlike primary sepsis. Most patients have fever and leukocytosis, can be cultured from skin lesions, occasionally blood. The good news: prompt antibiotics and surgical debridement are usually curative, much lower mortality than primary sepsis.
Finally, let's touch on Vibrio alginolyticus. What kind of infections does this species cause, and how serious are they generally?
V. alginolyticus was first identified as a human pathogen in 1973. It occasionally causes eye, ear, and wound infections, especially wounds contaminated at the beach. While severity varies, V. alginolyticus infection tends not to be serious or life-threatening in otherwise healthy individuals.
So generally less severe than vulnificus.
Generally, yes. It responds well to antibiotics and drainage if needed. Specific cases include otitis externa, outer ear, otitis media, middle ear, conjunctivitis, eye. Tetracycline treatment usually results in cure. It's also a rare cause of bacteremia in immunocompromised hosts, similar to other Vibrios in vulnerable populations.
Wow. What an incredible, in-depth journey we've just taken. Truly a deep dive into the profound complexities of cholera and other Vibriosis. We've covered everything from the most feared global pandemics and their historical impact to the intricate microscopic mechanisms of toxins. the nuanced differences between specific Vibrio species, and the critical importance of public health interventions. This truly feels like we've achieved a mastery-level understanding.
It's been quite the exploration, and I agree, a true journey to mastery. It reinforces the idea that understanding these pathogens from their microscopic structure to their profound global impact is fundamentally key. We've seen how precise details, like the molecular action of cholera toxin irreversibly upregulating adenylate cyclase, or specific environmental conditions like water temps over 20 degrees C for Vibrio proliferation, make all the difference in informing effective prevention, guiding rapid diagnosis, and ensuring life-saving treatment. The interconnectedness of biology, environment, public health policy, and individual clinical outcomes is just undeniable and beautifully illustrated by these organisms.
Absolutely. We've traversed from understanding the deadly rapid progression of hypovolemic shock in cholera to appreciating the nuanced differences between Vibrio species causing vastly different syndromes. From gut-wrenching gastroenteritis to life-threatening soft tissue infections and sepsis, and the complexities of vaccine development, from established killed oral vaccines to promising single-dose live attenuated strains, paint a picture of ongoing global efforts and remarkable scientific ingenuity.
What's fascinating here is how these granular details truly contribute to a professional level understanding. They enable informed decisions in medical practice, robust public health policy, even personal travel planning. The ability to connect an environmental factor like ocean temperature to disease incidence or a specific molecular mechanism like toxin action to a clinical symptom is what truly builds mastery and allows for effective intervention.
So as we wrap up this incredibly detailed deep dive, here's a final provocative thought for you, our listener. Given the remarkable adaptability of Vibrio species, their proven ability to acquire new virulence factors through lateral gene transfer, and the persistent challenges facing global health infrastructure in a rapidly changing climate, what further innovations, perhaps beyond what we know today in water sanitation, or what groundbreaking advancements in rapid diagnostics, do you think will be most critical in preventing the next major cholera outbreak or controlling the rise of other vibriosis in our interconnected world? It's a complex interplay, and the solutions will undoubtedly require ingenuity, collaboration, and constant vigilance. Thank you for joining us on this incredibly detailed deep dive into the world of cholera and other vibrioses. We truly hope you feel not just well-informed, but genuinely empowered by this deeper understanding of these fascinating, albeit dangerous, microorganisms. We'd love to hear what stood out to you from this journey.